I just ate a bunch of spicy stuff. Okay. Starting out, Phys and Histo, and the study guide, and immunology. Uh-huh. Phys and Histo shouldn't take too long. Okay. I'm pretty much I think I'm just gonna do the study guide stuff. I don't know if I look like a serial killer. I just ate some spicy foods. That's why my lip is so red. Hello. Sorry, I'm gonna get to the reviewing in a second. This will probably be pretty boring for a lot of people, but I'm downloading study guides right now. That's crazy. Okay, so bunch of cell stuff, talking about um, DNA, RNA, all that stuff, molecular biology, cellular basis of physiology. Man, how far down does this go? Ooh. Okay, lipid bilayer, phospholipids, cholesterol, glycolipids, peripheral proteins, integral proteins. Yeah. So why do the organs work? How do they work? Here, let me put this here now. How the cell functions. That's what we're going to be talking about on this thing. Yeah, the body has fluid, which is water and also extracellular fluid. Cytoplasm, plasma membrane. Okay, plasma membrane is not generally permeable. Hello, palm tree. Alright. Contains transporters like integral proteins, I think. And it's also a bilayer. It means it's got like 2 layers, I think. I don't know. I learned all this years ago. And I wasn't really paying attention during the lectures this past week. But plasma membrane is a lot of lipid, a lot of protein. It's about 50-50%. All of the lipids in a cell membrane are amphiphilic, which means they're hydrophobic. Wait, sorry. They've got a hydrophilic end and a hydrophobic end. So a hydrophilic end and a hydrophobic end, which means water-loving and water-hating end. Yeah, that makes sense. I mean, we learned that in high school. Fatty acid is a carboxylic acid with a long tail. Yeah. Phospholipid, glycolipid, cholesterol, sphingomyelin. Okay. Yep. Phospholipids. Van der Waals forces. Lipid bilayer. This is so boring. I don't want to go through all this. Yeah, I think I'm just going to go through the study guide. Honestly. Okay, so basically the membranes are made of lipids and proteins, phospholipids, glycolipids, and cholesterol. Cholesterol is only in mammals. All of those lipids are amphiphilic molecules. Again, have a hydrophilic and a hydrophobic end. the groups attached to phosphate and phospholipids. Yeah, what? Oh, those are head groups, apparently. Fatty acid tail is attached to the polar head via glycerol. Presence of double bonds in the tail determines whether the fatty acid tail is saturated. Saturated means it doesn't have double bonds. Unsaturated means it has a double bond. Sphingomyelin is made from sphingosine instead of glycerol, and it's polar. A free hydroxyl group contributes to polar properties found in the myelin sheath of a nerve. Okay. Glycolipids are found exclusively in the non-cytoplasmic monolayer, or the extracellular side of the lipid bilayer. Sugar-containing lipids like galactose, sphingosine, and the fatty acid tail. Wait, what? I don't know. Present in the nerve cells. Cholesterol is a sterol. Cholesterol. Yeah, that makes sense. Sterol with a rigid ring structure has a polar hydroxyl head group and a short nonpolar tail. Polar hydroxyl group orients closer to polar groups of phospholipids, resides between phospholipid molecules, absent in prokaryotes, very limited in the intracellular membrane, i.e. the membrane surrounding intracellular organelles, and it's enriched in nerve cells. It's energetically favorable interaction What? Oh, an energetically favorable interaction is key to bilayer formation in water. Okay. Hydrophilic regions are water soluble due to the presence of a polar group that can form hydrogen bonds with water molecules. Yeah, that makes sense. Hydrophobic regions are insoluble in water because they're nonpolar. Yeah. Spontaneously orient to bury hydrophobic tails in the interior and expose hydrophilic heads to water. Cholesterol can easily flip across the membrane due to a small polar head group, for example, OH. Phospholipids can move laterally, rotate, and flex, but can't flex due to the large polar head group, requiring a lot of energy to cross the middle hydrophobic region. Okay, the plasma membrane is considered a fluid mosaic. Fluidity depends on its temperature and composition, tail length, cis double bonds, etc. Shorter fatty acid tail or a bend in it due to the presence of double bonds in fatty acid tail, the unsaturated fatty acid tail. And this allows for more free movement. Okay, so the plasma membrane moves well. Phospholipids can move laterally, rotate, and flex, but it says they can't flex. That doesn't make sense. They just said they could. Whatever. Anyway, the plasma membrane, lipid bilayer, rigid, straight, saturated fatty acid, long chain, absence of any double bond in the fatty acid chain restrict the movement due to tight packaging/interaction among phospholipids. The rate of lipid movement depends on temperature. When diffusion occurs, lateral movement, the lipid is in the sol state, solution or liquid state. At high temperatures, the thermal energy of any given lipid molecule is greater than the interaction energy that would tend to hold adjacent lipid molecules together. With low temperature and minimal movement, it is in the gel state, between liquid to solid. So the lipid is in that state. Yeah, I mean, this makes sense. At high temperatures, fat melts. At low temperatures, it freezes, kind of. Membranes are fluid such that components can move about within the plane. Cholesterol, due to rigid sterol rings, maintains membrane fluidity by restraining the movement of phospholipids. This makes the lipid less deformable, and cholesterol decreases the permeability of the bilayer to water-soluble molecules and prevents freezing. Endocytosis is the process of capturing a substance or particle from outside the cell by engulfing it with the cell membrane and bringing it into the cell. Yeah. Exocytosis, basically releasing contents. Caveolin forces inward curvature of a membrane. Helps in the endocytosis by forming a cup-like structure around the vesicle. Flattening of caveoli allows the plasma membrane to expand in response to various stresses. Membrane fusion during neurotransmitter release at a synapse is mediated by V-snare, T-snare proteins, and SNAP proteins. Okay. Membrane proteins. Lipids provide structure, but membrane proteins perform most important functions. Membrane proteins can be peripheral, which means loosely bound, or integral, which means covalent attachment. Peripheral proteins form loose interactions with the integral protein via non-covalent interactions. They can be knocked off a membrane by ionic effects like salting out, which means it's precipitating protein. The hydrophilic portion of protein forms hydrogen bonds with water and other molecules in the membrane. Addition of salt changes the ionic salt concentration and may result in protein's precipitation. So these integral proteins are in the plasma membrane. All the proteins are amphiphilic, and a lot of them are glycosylated and referred to as glycoproteins. Detergents are amphiphilic as well, and they can solubilize proteins. The hydrophobic end binds to the hydrophobic region of the protein that displaces it from the membrane, and hydrophilic end interacts with water to solubilize them. An example of a peripheral protein is ankyrin. The biconcave structure of red blood cells is maintained by cytoskeletal spectrin dimers. Hi. Spectrins are linked together into a... Band 3 and glycophorin transmembrane protein are integral proteins. Okay. Not sure what all that mumbo jumbo was about. I got even more here. Diffusion through the lipid bilayer depends on the hydrophobicity of the molecule. More hydrophobic molecules can easily diffuse from a membrane. Strange language here. Uncharged polar molecules can also go across a bilayer as long as they're small enough and as long as there's not too much of them. Okay, the cell membrane generally doesn't allow passage of polar molecules because it's hydrophobic on the inside. But there's ways that cells still get stuff through because of transmembrane proteins. Osmolality describes the number of osmotically active solutes in a solution. Ugh. I don't even want to look at that. Okay, when something's isotonic, that means the osmolality is the same as that of a reference solution. Hypertonic has a higher osmolality, and hypotonic has a lower osmolality. Osmosis is the diffusion of water. Yeah. It has to do with isotonic and all that stuff. And then if you're dehydrated, you can give isotonic solution. If you have too much sodium in your body, you can give a hypotonic solution. That'll dilute the concentration. And wow, if you're in shock, you can give a hypertonic solution. Wow, okay. Cells can recover from shrinkage by opening certain sodium and chloride channels. Aquaporins form specialized hydrophilic transmembrane channels for water passage. This is where sweat, saliva, and tears secrete water through. Aquaporins. A bunch of electrical jargon here. Not sure exactly what it's saying. And okay, there's active and passive diffusion, simple diffusion, facilitated diffusion. Yeah. The rates of movement can change. Transmembrane proteins can move polar solutes across that non-polar membrane, yeah. Channels allow the ion to flow down the electrochemical gradient. In an ion channel, a transmembrane pore is either open or closed. GLUTs are the specialized receptors that mediate glucose transport down the concentration gradient. In type 1 diabetes mellitus, you can't release insulin, and so that results in low rates of glucose uptake into tissue. And so your blood glucose is going to be high after carb-rich meal. Vasopressin, transporters, chloride bicarbonate exchanger of antiport and electroneutral exchange. active transport. Yep, ATP driven. An example of that is the transport of the sodium and potassium pump. Sodium, potassium pump, 3 sodium out and 2 potassium in. Uses ATP. There's 2 types of active transport, primary and secondary. Primary energy from ATP hydrolysis drives a solute against an electrochemical gradient. And then secondary, a gradient of an ion has been established by primary transport. And it moves it down the electrochemical gradient. The movement of that solute down the gradient provides the energy to drive a second solute down a gradient. An example is glucose and sodium symport on the intestinal cells. Okay, P ATPase, V ATPases, nucleoside binding domains, uses ATP, chemotherapeutic drugs, doxorubicin, and vinblastine. Okay, potassium leak channels, sodium potassium ATPase, membrane potential, ion channels, closed, open, and inactivated voltage gates, voltage gated sodium ion channels. Acetylcholine receptor, allows sodium, calcium, and potassium to pass through with ease, but other cations and anions are unable. GABA, glycine receptors, endocytosis, vesicular phagocytosis. Drug absorption, lipophilic and unionized drugs are easily absorbed as compared to hydrophilic and ionized. Oh, unionized. Looks like unionized. pKa is the pH when the drug is 50% ionized and 50% unionized. A drug that is a weak acid will be best absorbed in an acidic environment, least ionized in an acidic pH. A drug that is a weak base will be least absorbed in an acidic environment, least absorbed, and a drug that is a weak acid will be best absorbed. So a weak base will be least absorbed. Okay. Weakly basic drugs are more likely to be absorbed in alkaline environments. I don't know, doctor or engineer? It's a good question. Depends on which you would rather do. Would you rather build, I think, and engineer, or would you rather do surgery, have patients, have clients? You got to deal with a lot of people as a doctor. As an engineer, maybe not so much. I'm not sure. But yeah, it just kind of depends on what you would prefer, I guess. You can do both. I don't know. Which one would you rather do, I guess? Or just pray about it. Ask God. He'll tell you. Weakly basic drugs are more likely to be absorbed in alkaline environments, which is a high pH environment, where they remain unionized compared to acidic environments. Okay, and the other way around. To assist excretion of an acidic drug via urine, alkaline environment is favorable. Okay, that was boring. Now, onward to immunology again, I think. Let's see. Yep. Okay, excuse me. So, the spleen is an abdominal organ. Histologically, it has red pulp and white pulp. Red pulp basically involves blood filtration, and it's the most of the spleen. White pulp is immune role. It's formed by lymphocytes surrounding the arterioles running through the spleen. The red pulp contains RBCs, which are red blood cells, platelets, and macrophages. Okay, and then there's also periarteriolar lymphoid sheaths, or PALs, P-A-L-S. Those are rich in T cells. Then there's marginal zones, which have macrophages and a type of B cells in them. Then there's lymphoid follicles, which are rich in B cells. Goodness, how much of this is there? Oh, not that one. It's okay. Let's go. The spleen is not connected to the lymphatic system. It filters antigens coming from the blood, which is important for blood-borne pathogens. The marginal zone surrounds the follicles. It has few T cells, is rich in macrophages, and has resident non-circulating, a resident non-circulating population of B cells known as marginal zone B cells. These B cells rapidly produce antibodies that have low affinity to bacterial capsular polysaccharides. Okay, these antibodies provide some degree of protection before the adaptive immune response is fully activated. It's not foolproof, though. They don't recognize proteins, for example. Blood-borne microbes and soluble antigens and all that are filtered from the blood by macrophages and immature dendritic cells. Okay. And those dendritic cells go to the T cell areas after grabbing an antigen. There, they can present the antigens to the T cells. MALTs, mucosal associated lymphoid tissues. There's many lymph nodes that antigens are drained into. Okay, review. DAMPs, damage-associated molecular patterns, are signals released by damaged cells to alert the immune system. And they are also host-derived. They come from the host cells. Neutrophils are the most abundant type of white blood cells in circulating blood and play a key role in innate immunity. Bacterial infections activate neutrophils more often. Okay, secondary lymphoid organs, the tonsil and the lymph nodes. Primary, the fetal liver, thymus, bone marrow, and the bursa of Fabricius. Okay. Matching. MALT means mucosal associated lymphoid tissue. Peyer's patches are in the small intestine and especially the ileum. Autocrine means the cell produces and receives the signal. A lymph node has a cortex region with discrete B and T cell areas. Adaptive immunity is correlated with memory. Endocrine is hormones. Paracrine, it's the cells that signal other cells that are nearby. Innate immunity is fast response and always present immunity. Thymus has a lobular structure and it is involved in T cell development. Okay, and then there's antibody structure. There's like the antigen binding site, variable regions, light chains, heavy chains, constant regions. Yeah. Okay. That's pretty much it. Pretty much it for today, or that day, the 25th. I don't think we did anything in anatomy. Let me check. I don't think so. Nope. Nope. Nothing. Okay, well, I guess that's it for this recording. And, all right. Thanks for watching. See y'all later.